Hello, good evening. So this is a continuation from the previous session where we discussed the glycolytic pathway in study session 1 and 2, as you can see here. All right, so for study session 3, we're talking about the tricarboxylic acid cycle. All right, based on your course material, we're going to discuss the description. We're going to describe the cycle, the amphibolic nature of the cycle, and aplerotic nature of the cycle, the relationship between the citric acid cycle or TCE cycle and beriberi, which as nursing students should know this, right? Summary of oxidative phosphorylation, inhibitors of the electron transport chain, which I'm sure I've highlighted in the previous classes. All right. Okay. So in eukaryotes, like you and me, the TCA cycle takes place in the mitochondria. Okay, because all the enzymes of this cycle are located inside the mitochondria. And we all know that the mitochondria is called the powerhouse of the cell. The glycolytic pathway usually takes place in the cytoplasm, right? But the TCA cycle takes place in the mitochondria. Why? Because the mitochondria houses the enzyme that catalyze the reactions of this pathway. So I think it makes sense now, right? The TCA cycle is an important source of precursors or building blocks for the synthesis of molecules such as amino acids, purine bases, cholesterol, and porphyrins. So to show this in a picturesque manner, I will take you to this slide here to make sense of what I'm actually saying. So basically, this is the TCA cycle. If I share this screen, so you see clearly, okay? This summarizes everything. Yes. Now, this is your glycolytic pathway. Glucose enters into your cell, maybe after eating a meal or something, right? This is your cytoplasm here, where the glycolytic pathway takes place. And this is your mitochondria, where your TCE cycle takes place. Now, if you remember a glycolytic process very well, 10 enzymes are involved, which of course you should know. Glucose is converted to glucose 6-phosphate by hexokinase. Glucose 6-phosphate can also go into a pathway called the pentose phosphate pathway, which is not part of this lecture, so we're not going to discuss that. Glucose 6-phosphate is converted to fructose 6-phosphate in the glycolytic pathway by an isomerase, right? This fructose 6-phosphate can go into the hexosamine pathway, which is a very important pathway in some diseases like diabetes, for example, and even cancer, hexosamine pathway, right? Fructose 6-phosphate is converted to fructose 1,6-bisphosphate by phosphofructokinase 1, if you remember very well. And we said this reaction here is irreversible, just like your conversion of glucose to glucose 6-phosphate is irreversible, as well as conversion to pyruvate is also an irreversible. Phosphoenol pyruvate to pyruvate is also irreversible, right? And I remember very well telling you that conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase 1 is the rate limiting step of the glycolytic pathway. It's a point of no return. Glycolysis has to occur if it reaches this stage. Why? Because although this first reaction here from glucose to glucose 6-phosphate is irreversible, but glucose 6-phosphate can go into pentose phosphate pathway. So glycolysis might not occur, right? Fructose 6-phosphate can go into the hexosamine pathway, so glycolysis might also not occur. But once fructose 6-phosphate is converted to fructose 1,6-bisphosphate, That's the rate limiting step. Then glycolysis is most likely going to occur, right? Now let's skip all this. Let's come to this second phase of glycolysis, the P-off phase. Now this is 3-phosphoglycerate. Now 3-phosphoglycerate can be converted into amino acids such as serine, glycine, or can participate in 1-carbon metabolism. So this is what your lecture note was saying. Intermediate of the citric acid cycle can serve as a building block. Building blocks for can serve as precursors or building blocks for synthesizing macromolecules and things like that, or amino acids or lipids, for example. Now, this is just an example from the glycolytic pathway. Now, if you go into the citric acid cycle, come down under aerobic condition, just like in mammals, you and I, your pyruvate is converted to acetyl-CoA by pyruvate dehydrogenase complex, right? Acetyl-CoA condenses with oxaloacetate here to produce citrate. Citrate is converted to isocitrate, alpha-ketoglutarate. Keep going. This is just a summary. We'll go into the detail. Now, these are intermediates you get in the course of the TCA cycle. Citrate can be converted to fatty acid or sterols, right? Alpha-ketoglutarate can be converted to amino acids such as glutamine, glutamate, or proline. Succinyl-CoA can be converted to heme. Heme is a component of your hemoglobin, right? Oxaloacetate can be converted to amino acids such as asparagine and aspartate. So I think this makes sense now. The statement here has been substantiated using this simple illustration, right? Okay. The cycle starts when a 4-carbon compound oxaloacetate condenses with a 2-carbon acetyl units of acetyl-CoA to yield a 6-carbon tricarboxylic acid citrate. So usually you read this, but if you look at the cycle and study it, it's easier to just understand everything. So instead of me reading this, I'm just going to explain the cycle. Then you take your time and read on your own, if that's okay. This is the cycle, basically. This is your TCA cycle. Your acetyl-CoA, which comes from conversion of pyruvates, right? The pyruvates from the glycolytic pathway is converted to acetyl-CoA. Of course, you know the enzyme involved. Okay. This resolution is not so good. So let me see if I have an illustration with a better resolution. I think that will go a long way. Okay. Okay. Yes, this is the TCA cycle, tricarboxylic acid cycle. Yes, this is very good. So summarily, this is what we have. But this one, I think this is perfect. Let me just present this. All right. Now, isn't this interesting? This is your acetyl-CoA. It condenses with oxaloacetate here in the presence of citrate synthase to produce citrates. If you see, you have a 3-carbon compound here plus another 3-carbon substrate here to produce a 6-carbon substrate, which is citrate. Citrate is converted to isocitrate, which is an isomer of citrate. The enzyme that does this is aconitase, and the catalysis occurs in 2 steps. First, aconitase dehydrates citrates and then rehydrates the cis-aconitate intermediate. So basically, it's just an isomerization reaction occurring here. The enzyme that does this is aconitase. All right. And then your isocitrate now is converted to, is decarboxylated to alpha-ketoglutarate. Decarboxylation means removal of CO2, a molecule of CO2, right? And you have alpha-ketoglutarate. And if you remember the previous figure I showed you, alpha-ketoglutarate can be used to produce, can serve as a precursor for synthesis of proteins through the synthesis of amino acids such as glutamine and glutamic acid, right? If you remember very well. Now, your alpha-ketoglutarate in the citric acid cycle is further decarboxylated to give you succinyl-CoA, right? CO2 is released, and you have succinyl-CoA. CoASH is added. The enzyme that does this is alpha-ketoglutarate dehydrogenase complex. All right. Your succinyl-CoA is now converted to succinate. The enzyme that does this is succinyl-CoA synthetase. And it requires, it leads to the production of your energy currency, ATP. ADP goes in, ATP comes out. This type of ATP generation is called substrate level phosphorylation. It does not require your conventional NADH coenzyme, right? Your succinate is produced. Succinate dehydrogenase converts succinate to fumarate, right? And this is the other coenzyme I mentioned. I think if I remember very clearly in my previous classes, you know about NADH. There are reactions that require NADH. There are reactions that require FAD. And you have NAD and FAD, right? And you end up with FADH2. Now, this FADH2 and NADH are used up in the electron transport chain to generate ATP. NADH generates 2.5 ATP. FADH2 generates 1.5 ATP, okay? Fumarate is hydrated to form malate, while malate is dehydrogenated to form oxalate. So basically, your oxaloacetate is recycled through the pathway. It condenses with this. By the end of your reaction, your oxaloacetate is regenerated. Okay. Now this summarizes everything. Your Krebs cycle. Oxaloacetate condenses with acetyl-CoA to get citrates. Citrates is isomerized. Isomerizes to isocitrate. Isocitrate is decarboxylated to form alpha-ketoglutarate. Now, this reaction is catalyzed by isocitrate dehydrogenase, and it produces NADH, which is going to go into the electron transport chain. Alpha-ketoglutarate is converted to succinyl-CoA. The decarboxylation reaction produces NADH, which is also going to go into the electron transport chain. Succinyl-CoA is converted to succinate. This is substrate level phosphorylation. So we have one molecule of ATP produced from one molecule of acetyl-CoA, right? Succinate is succinate dehydrogenase is dehydrogenated to fumarate. FADH2 is produced, which yields 1.5 ATP, right? It goes into the electron transport chain, like you see here. Fumarate is hydrated to malate, while malate is dehydrogenated to oxaloacetate, which also produces NADH coenzyme, reduced coenzyme, which also goes into the electron transport chain. And now let's count. You have NADH2, 1, 2, 3. Okay? NADH, 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 right? And then you have FADH2 here. Products, we have ATP here. Now, you take the products of this reaction and those from the glycolytic pathway, and you put everything together. I have to juggle your memory so you understand what we are actually saying here. If not, there's going to be a confusion. Okay? This here is your glycolytic pathway. In the glycolytic pathway, fructose 1,6-bisphosphate here was broken up into, let me use the biochemical term, was lysed by aldolase. Or lysed means just hydrolyzed by aldolase into glyceraldehyde 3-phosphate dihydroxyacetone phosphate. But dihydroxyacetone phosphate was converted to glyceraldehyde 3-phosphate. So we ended up with 2 molecules of this. And because we have 2 molecules of glyceraldehyde triphosphate, we are going to have 2 molecules of this, and 2 molecules of pyruvate. So what's entering the citric acid cycle is not just 1 molecule, but 2 molecules of pyruvate. 2 molecules of pyruvate will be converted to 2 molecules of acetyl-CoA. If you agree with me, right? Now, 2-NADH is produced from the glycolytic pathway, and 2-ATP is produced from the glycolytic pathway, plus 2 pyruvates. All right, so now, you see, 2 pyruvate. There has to be a balance, 2-acetyl-CoA, right? So the production of acetyl-CoA from pyruvate yields NADH. Now, since 2 molecules of pyruvate was produced from glycolysis, we expect 2 molecules of acetyl-CoA and 2 molecules of NADH, right? So we already have 2 molecules of NADH from glycolysis, plus these 2 molecules here. We now have 4 NADH, okay? All right. So now the acetyl-CoA will go in. Let's see, 1 acetyl-CoA gives you 1, 2. Okay, let's come in here and let's do this practically. So it makes sense. You see, 3 NADH was produced from 1 molecule of acetyl-CoA and 1 FADH2. Okay, all right. Now, my assignment to you, if you're listening to this lecture, is I want you to give me the total number of NADH and FADH2 produced through the glycolytic and the TCA cycle. This is your assignment. I need you to do this for me. And what molecule of ATP? How many molecules of ATP should we expect from this? I think I will make it a written assignment in the forum. After listening to the lecture, you should respond to that. Okay? All right. So basically, I think I've touched on almost everything in the TCA cycle. You can read the lecture note on your own. Mention the 2 compounds that begin the Krebs cycle. It's oxaloacetate and acetyl-CoA. Now, the amphibolic nature of the TCA cycle is the citric acid cycle functions as a catabolic pathway when it is used to break down acetyl-CoA to 2 molecules of CO2, water, and energy. So whenever the energy of the cell is low, right, the catabolic pathway is favored because you need energy. So the TCA cycle will go full force towards the production of NADH, FADH2, ATP, and it will go into the electron transport chain to produce more ATP. All right, the cell cycle functions as anabolic. On the other hand, the TCA cycle, sorry, can also function as an anabolic pathway when there's sufficient energy. You don't need ATP. The body operates on the principle of maximum economy. If there's no need for energy, you don't need to be producing ATP. So the precursors or the intermediates that could be used to synthesize other things like fatty acids, amino acids, sterols, and things like that from the citric acid cycle will go in that direction instead of the direction of ATP production, right? I think it makes sense. While for the anaplerotic nature of the cycle, what do we mean by anaplerotic nature? The ability of the cycle to replenish lost intermediates, right? More of salvaging, replenishment. So the TCA cycle has a single substrate, and the substrate is acetyl-CoA. So in most cells, however, there's a considerable withdrawal and addition of intermediates into and out of the cycle. Because, say, you want to replenish, you want to kickstart the TCA cycle, but not through the glycolytic pathway, amino acids can be converted to intermediates of the citric acid cycle, and the cycle will continue. Wouldn't you agree? For example, alpha ketoglutarate produces amino acids like what do you call it? Glutamine or glutamates, for example. So it means glutamine or glutamate can be converted back to alpha ketoglutarate, which is an intermediate of TCA cycle. Oxaloacetate can be converted to amino acids such as asparagine and aspartate, for example. So it means if we don't have enough glucose and you have enough amino proteins that could be hydrolyzed into these individual amino acids, aspartate or asparagine can be converted by the cell to oxaloacetate, which will now condense with maybe acetyl-CoA that comes from fatty acid catabolism or something. You understand? So looking at things from a holistic perspective, it makes sense, right? So like it's mentioned here in your lecture note, oxaloacetate and alpha ketoglutarate are used in the synthesis of several amino acids. Citrate is the source of acetyl-CoA in the cytosol, right? Citrate can be converted to acetyl-CoA. Basically, that's what this statement is saying, which is used for the synthesis of fats, other lipids, and some amino acids, which I've shown you in my slide. Okay, so you can read the rest. I think this is self-explanatory. The relationship between TCA cycle and beriberi. Okay, so what is beriberi? Right? Okay. This is very important since you are nursing students. We should relate the biochemistry to some disease, right? So you see the practicability of what we are teaching you. Beriberi is a neurological and cardiovascular disorder which is caused by deficiency of thiamine, also called vitamin B1. Vitamin B1 is thiamine, right? I think vitamin B2 is what? Riboflavin. I stand to be corrected. B3 is what? I think niacin. B4 is what? You should know. Um, what is vitamin B5? That's pantothenic acid, if I'm not mistaken, right? Vitamin B12 is, I think, cobalamin. I'm not very sure, I've forgotten. Or you should know, right? Nurses. All right. The disease is characterized by, so a deficiency in vitamin B1 causes beriberi. Now, why are we talking about this in the TCA cycle? That's the question you should be asking. Okay. So because thiamine pyrophosphate is a prosthetic group, it's required by a certain enzyme that catalyzes the conversion of pyruvates to acetyl-CoA. The absence of this enzyme, of this prosthetic group, or call it a prosthetic group, can be a coenzyme or cofactor. They are compounds or molecules required by enzymes to function properly. We call them prosthetic groups. So thiamine pyrophosphate is an example of such. This enzyme, pyruvate dehydrogenase, and even alpha ketoglutarate dehydrogenase in the citric acid cycle require TPP, thiamine pyrophosphate. So deficiency of this causes beriberi. So basically, what we are saying here is a TCA cycle. The absence of this enzyme can compromise your TCA cycle, and that's where you have patients with this form of disorder called beriberi, deficiency in thiamine pyrophosphate. So I think I'll just show this. I think I have it here. Exactly, this is it. So this prosthetic group, TPP, is thiamine pyrophosphate. It's required by pyruvate dehydrogenase complex and alpha ketoglutarate dehydrogenase, the enzyme that catalyzes the conversion of alpha ketoglutarate to succinyl-CoA in the citric acid cycle. So I might ask you a question. I think I've asked one question already. So this is also very important. Pyruvate dehydrogenase complex, alpha ketoglutarate dehydrogenase. So I need you now to tell me the prosthetic group that is important for this two enzymes. The absence of this prosthetic group causes beriberi. What is the prosthetic group? What's the name? Thymine pyrophosphate, derived from what? Vitamin B1. So you should know this, right? I think it's very straightforward. Right? When you're looking at this reaction, you see the pyruvate dehydrogenase complex requires NAD plus, which is a coenzyme. It requires FAD, which is a coenzyme. Requires lipids, which is a compound, lipid compound, and then it requires thymine pyrophosphate. Okay, so I think basically this is it. That's the end. And then the NADH, FADH2 generated from the glycolytic pathway end up going into the, what do you call it? The electron transport chain, which I think you could also see here. All right. Just roughly. It's part of your mitochondria. This is your mitochondria, right? Yes. All right. Okay, where in the electron transport chain, NADH, sorry, I keep saying NADH2, it's a mistake, it's an oversight. I'm sorry about that. NADH produces 2.5 molecules of ATP, while FADH2 produces 1.5 molecules of ATP. So basically, this is what happens. And the process that produces these molecules of ATP in the electron transport chain is called oxidative phosphorylation. All right. Now, inhibitors of the electron transport chains, So, of course, you should be able to regulate things, right? Say in cancer cells, mostly in cancer cells, anaerobic glycolysis occur, really. The cancer cell usually end up uh, evolving. So the inhibitors of the electron transport chain include the barbiturate drugs, antibiotics, and insecticides, especially those that are selective. So example of barbiturate drugs include, of course, amital. I think you should know this better than me. Antibiotics have different types of antibiotics. Of course, not all antibiotics will inhibit the electron transport chain, but some will, right? Okay. Okay, so this is an ITQ question for you. Beriberi disease is characterized with dash and dash symptoms, neurologic and cardiac. You should take your time and attempt these questions. Okay, it's going to be very helpful to you. All right, this will be the end of study session 3, module 1. Thank you for your kind attention. Okay.